All right, today we're going to talk about the next area of the study design, which is really all about our disease and our immune system. It's a very, very interesting area of study design, really relatable. It's also quite a difficult area of the study design, just in terms of the amount of content, but because we can really relate it to our own lives and it helps us understand our own diseases a bit more, I find it very much an interesting area. So today we're going to look at these things that we call pathogens and their antigens. Okay, so it's chapter 6 in your Nature or Bio books. And an example of a pathogen is a virus, such as, I don't know if you've heard it, but the Zika virus, all right, which was a virus, is a virus that is a huge issue in tropical countries, and especially, for example, in South America, Brazil, where Mr. Mello is from. So here we've got the Zika virus. Great, I'm not dating Mosquito Girl again because the Zika virus is passed by mosquitoes. Hilarious. Have a go at these 3 quick questions and we'll have a look at them together in class. All right, so in the study design, we're in this topic of responding to antigens. Okay, we'll talk about what that means in a bit, but an antigen is a unique special molecule or a part of a molecule that actually starts a immune response. So antigen, it literally means antibody generator. Cool. Hopefully you've heard of antibodies before. If not, we'll talk about heaps about them later. And so we want to be able to create a distinction between non-self antigens, self antigens, and allergens. We're not going to talk about allergens today, but we are going to talk about non-self and self antigens. And then also we have to talk about invading cellular and non-cellular pathogens. There's a few examples we'll go through as a source of non-self antigens and some preventative strategies including physical, chemical, and microbiological barriers in animals and plants. We're not going to really talk about plants, but I've got a great video for you to watch about this. Okay. So that Zika virus that we're talking about, it actually gets transmitted by this specific mosquito. Okay, and if you click this picture, it actually has a link to a news story about this mosquito and the Zika virus. And this mosquito is called the Aedes aegyptus mosquito. I think I spelled that right. And this mosquito is endemic to Africa. Okay, but it was eventually spread to South America, and even now it's in north of Australia. Okay, and this mosquito is very important because it's sort of spreading this virus, and we're going to talk about what a virus is and what the spread of a disease such as this, how it happens. Okay. But first of all, what is a disease? A disease is simply any conditions that impairs, so it obstructs the normal functions of an organism. So if you break your leg, in theory, you have a disease. If you get a cold, obviously a disease. Okay. The difference between those two diseases is that if you break your leg, you're not going to pass it on to the person next to you. If you get a cold, you might pass it on. And that's how we categorize diseases into infectious, so diseases that can pass on, and non-infectious diseases that can't pass on. All infectious diseases involve the invasion of a non-self invader, which we call a pathogen. Okay? So, pathogens are disease-causing agents. All right? We can't say they're disease-causing organisms because not all pathogens are actually alive. All right? So disease-causing agents that come in lots and lots of different forms. Here's some example of these that we'll go through. All right, so the first example is viruses. They're probably the coolest pathogens. They're actually non-cellular, so we consider them not alive. And they're composed of a protein coat and some nucleic acid on the inside. And this nucleic acid can be either DNA or RNA. They're an obligate parasite, which means that the only way that they can actually survive is by infecting something and living inside of that and reproducing through that infection and then going on and infecting other things. And they actually act by either inserting their own DNA or RNA into the DNA of the host cell. Okay, so it directly attacks the DNA of that host cell. And because of this, the host is actually going to transcribe and translate new viral particles, new viral proteins, which are going to assemble themselves into brand new viruses. That's how they reproduce. Okay. Later on, these new viruses are then released in the extracellular space when the host cell breaks, which means this is what lysis means. And watch this. This is a virus entering the cell. Okay, so you can see there, and it fuses with the plasma membrane, enters the cell. This is the viral genetic material that's released. The other purple stuff was some proteins. And then that virus, it's going to show us again, that viral nuclear material, viral DNA, is going to go and attack the nucleus and thus the DNA of the actual cell, do the stuff that we just talked about. All right, so this is a little figure of how a specific kind of virus called the bacteriophage works. It's quite an important diagram. We're going to go through this concept later on a little bit more in detail, so you don't have to stress too much about it for now. All right, the other type of pathogen that we'll talk about is called bacteria. All right, and you've probably heard of bacteria. They're actually the very first type of life that appeared on Earth. They're a single-celled organism, and it's simply the most abundant and diverse group of living things on Earth. All right, and only a tiny percentage of bacteria actually cause disease. For example, your hand is very full of bacteria. Go wash your hands. But your hands will still be very full of bacteria, and that's a good thing, okay? Most bacteria are good for us, but the bacteria that are bad for us can be really bad. Bacteria come in lots and lots of shapes, which give them their names. For example, bacilli, all right, I think of a baston, which is like a rod, okay? They're these kind of rod-shaped bacteria. Cocci, all right, I think of coconuts, because they look like coconuts. And spirally, I think of spirals. Okay, they're just 3 different type of bacteria, and we name them, for example, meningococcus. You see how that the end of that word is coccus, because they're this kind of bacteria and they attack your meninges. Okay, and that's the kissing disease, isn't it? What's it called? I forgot. Anyways, the general structure of a bacteria is sort of like this. Not all bacteria have all of these features, but every single bacteria has some DNA on the inside, and it's circular DNA because it's a prokaryote. All right. All bacteria are going to have a plasmid DNA, and we'll talk about the importance of the plasmid later on, but it's simply DNA that can get exchanged with other bacteria. All right. All bacteria have ribosomes because they produce their own proteins. They all have a plasma membrane, and they all have a cell wall. Okay. Some bacteria have what we call a flagellum, all right, which helps in movement, just like a sperm flagellum does. Okay. Some bacteria also have what we call these cilia or fimbriae, all right, and they're just sort of for protection or other functions. Okay. But that's just the general structure of a bacteria. Fungi is another kind of pathogen. They're both unicellular, so made up of one cell, or multicellular, lots and lots of cells, and they're eukaryotic. So bacteria were prokaryotic, viruses were non-cellular at all, and fungi are the first eukaryotic pathogen that we have. Most fungal infections of humans, they're external, they're very irritating, and they inflame the skin. Okay, they're usually not overly serious, but they are a nuisance. A great example of this is if you've ever had a little ring on your skin like this. It's called ringworm, which ironically isn't a worm at all. It's actually a fungus. Or if you've had, you certainly have, I think everyone in the world gets this at some stage in their lives, this sort of blistering and whitish between your toes, that's called tinea or athlete's foot. So that's tinea, and that one is ringworm. Both different kinds of fungal infections. Protists, another kind of pathogen. This time they are always unicellular, so they're only one cells, but they're still eukaryotic, and they can reproduce both sexually and asexually. Protists often spread diseases through what we call vectors, all right? And vectors are quite common in a few different kinds of pathogens, such as Plasmodium, which is the protist that spreads malaria, which you've probably heard about. It's probably one of the most important diseases in the third world. And malaria, so the Plasmodium protist actually goes and lives half of its life inside a mosquito, okay? And then a different kind of mosquito, the Anopheles mosquito. And then that Anopheles mosquito, when it actually bites you, right, it actually injects the larval stage of that protist in you, and then that protist go on to affect your red blood cells, such as this red blood cell here, and they grow into the full protist. And they go on, and then when a new Anopheles mosquito bites you, those new protists go into the mosquito, lay their eggs, and restart the process. All right, so now we've talked about a few pathogens, let's talk about how our bodies stop these pathogens, all right? And the first line of defense for our body, what we call the first line of defense, is the innate immune system, okay? So this involves a bunch of non-specific barriers, okay? These are just barriers that are complete barriers for your body, and they're going to try to keep everything out, okay? And these barriers are really just on the external surface of the body. We're going to talk about things that are in your mouth and in your gut, but technically that's still external. If you think about it, if you open your mouth, all right, your whole gut system, all right, all the way out your other end of your mouth, this system all is actually technically external, okay? Think of the first line defense like the in an NFL game, the defense at the very front, all right? They're going to stop the other team from getting to the quarterback, if you know anything about NFL. All right, so let's talk about some components of the first line defense. First components are physical barriers. Okay, these are things like intact skin. Cut skin doesn't work very well as a barrier, so it has to be intact. Ciliated mucous membrane. So, for example, in your trachea, which is your breathing pipe, you've got these little hairs that actually stop pathogens from getting in. You've got similar things in your nose. Expulsion reflexes, so things like sneezing, coughing, or physical barriers. We've got things like chemical barriers, such as antibacterial agents in your tears and saliva. We're going to run a little prac on this. Your stomach acid, very low pH. And then we've got things such as, oops, that didn't work. Look at that picture when the PowerPoint is still, maybe. We've got things like microbiological barriers. These are living things that work as barriers. So, for example, your blood clotting agents, your natural microflora, okay, that's the biggest one. So these are bacteria that live in your gut and they work as a barrier themselves. Have a go at actually going through this and working out whether it is a physical, a chemical, or a microbiological barrier here, okay? So the first one we have is the removal of particles by cilia in the nasopharynx. All right, so hopefully you recognize that that's a physical barrier. All right, the mucus lining in your trachea, we didn't talk about this one, but the mucus is a chemical. So number 2 is a chemical barrier. Your stomach acid, we said, chemical barrier. Normal flora, 3 is a microbiological barrier. Flushing of the urinary tract is a 1. That's an expulsion reflex. Rapid change in pH, that's another chemical barrier. All right, this one here we sort of split up because we have normal flora in our skin, so that's a microbiological barrier, but the intact skin surface is a physical barrier. The lysozyme in your tears are chemical barriers. All right. So last thing we're going to talk about is how our bodies actually do distinguish self from non-self. So in spite we having a very, very effective first line defense, it does a fantastic job. Sometimes pathogens do get in. All right, so it's important our bodies can actually recognize and kill these invaders, which is what we're going to talk about next time. But how we recognize these invaders are through markers. And what markers are, they're actually just receptors. The specific types of receptors on the surface of a substance or a cell that actually indicates where they're from. Okay, there are two different types of, and the two types are these: the MHC1 and MHC2 markers, and these compose of what we call the self markers. So they're the markers that tell our body that this cell belongs to us. Okay, MHC1 markers all cells have. Okay, MHC2 markers only specific leukocytes. We're going to start calling them leukocytes, but they're the fancy biological word for white blood cells. Okay, only very specific leukocytes have. Okay, what MHC1 and MHC2 markers stand for, you don't have to know this. You can just use the MHC acronym, is Major Histocompatibility Complex, which just means that it's the complex that your body uses to recognize itself. Okay, we'll talk about those later on. Non-self markers, on the other hand, so markers that your body sees that as don't belong to you anymore, the biggest group of them and the most important ones are those antigens we started talking about. And unfortunately for us, also non-self markers can be labeled as allergens. Okay, so how does that work? All right. This is a T cell, which is one of those examples of these leukocytes, all right? And T cells have these MHC2 markers on their surface, which is these, okay? Some of these MHC2 markers tell these T cells that it's a self receptor, okay? So if a cell has that, for example, this cell, it has a self antigen, and that self antigen, which is the MHC1 marker, by the way, you don't have to call it a self antigen, it's going to be attached to this T cell, and the T cell is going to be like, cool, that cell belongs to me, I'm not going to attack it. On the other hand, this T cell is going to have other MHC2 markers that recognize non-self. So if we have a non-self antigen, for example, it might be a bacteria or something, that bacteria is going to attach itself to the MHC2 marker, and the T cell is going to say, nah, I'm going to attack this cell.